Welcome back, everybody, to the Crosse Thinker Podcast. So today we have the guest Richard Mclaughlin to talk, who is teaching now at Gateway Technical College, and about natural science. And today our topic of the research will be two things. The first one will be the bacteria diversities in fecal materials of animals, so basically bacteria in animals. And then another one will talk a lot about the mammals and how to protect them, especially in Chinese Yangtze River. Okay, so let's start with, how did you get into biology from the beginning?
Biology, I've always been very interested in biology ever since I was a teenager and wanted to go to medical school. Then I went to undergraduate and I took a microbiology course with Professor Dr. Wool. I really liked the course, and I did a research project with Dr. Wool, and I said, This is very interesting.
What research did you do there?
I did just some basic E. coli research and trying to use a technique called electroporation to get plasmids into the E. coli bacteria. So it was a fun undergraduate project. And I said, well, this is good. I think I'm going to change. So what I did was I applied to graduate school. So I'm Canadian. So I applied to Queen's University. It's in a small city in Ontario called Kingston. And I worked with corkscrew-shaped bacteria called spirochetes. And at that time, they were creating a physical map. So what you would do is you would isolate the DNA from the bacteria, and you would cut it with enzymes called restriction enzymes. Restriction enzymes cut DNA. And then you would use a pulse field gel electrophoresis, separate out the fragments, size the genome. It was an okay technique. Now that technique has been completely replaced with genome sequencing and is used very, very little. So after finishing a master's degree, I went to McGill University, which is in Montreal, Canada, and worked with Professor Dr. Chan. Dr. Chan studied oral anaerobic spirochetes, and he studied Treponema denticola. He was actually the first person to discover the bacterium and publish a paper, and he looked at how the particular bacterium causes periodontal or gum disease. So we grew the bacteria, studied the bacteria, produced some papers on that. And afterwards, I worked for a company in Montreal, Neurochem, and we looked at this is different than microbiology. We looked at amyloid. So amyloid is a protein that aggregates in brains, especially in people with Alzheimer's disease. And we were trying to come up with a small molecule of medication to prevent the aggregation of the amyloid and as a possible treatment for Alzheimer's disease.
That's where your patent come from, right? Then I saw you have a patent.
Yes, I have
for treating aggregation disorders.
That's where the patent came from. Yes, so it was a different protein than the A beta. It's the alpha synuclein for treating Parkinson's disease. So another scientist and I came up with that idea with aggregation.
I see. Are people using those kind of treatments now?
Unfortunately, that patent expired. They did not renew it, and we tried a small molecule in clinical trials, and it was not successful. So the company took a different direction from the aggregation disorders. So unfortunately, it did not become a medication on the shelf.
Still a nice try, yeah.
It was a try. In clinical trials, approximately 1 in 20 medications that are tried become a medicine, so it's difficult.
Interesting, because I recently read a lot about Alzheimer's, and seems like when we get older and older, because people's lifespan becomes so much bigger now, that's going to become a major cause of a major disease older people have after they become like 60, 70, because people are just living longer in general now.
That's right. Yep. Number one cause of dementia, and there's treatments, but still not where scientists would like them to be.
Cool. So, let's first talk about your studies of bacteria in animals. You have a lot of publications on that. So, can you give us some interesting findings or research you have done before?
Yeah, my lab, so when I'm at Gateway Technical, I work with undergraduate students. And if you look in the literature, you're going to, with bacteria, find two major areas. Humans, so a lot is disease-causing with humans, and the other, if you look at animals, is some commercial application, so say cattle or swine, goats, horses, because there's commercial applications. Very little is done with the microbiota, the bacteria, other microorganisms associated with non-feed animals, non-commercial animals. So that's something that I'm interested in doing. So I've worked with Phil Cochrane. He was the chair of biology at St. Mary's University on the timber rattlesnake. So he would give me this particular snake unfortunately got run over by a lawnmower. We looked at the microbiota of the snake, and we published a paper on that. We also found in that particular sample a new species of bacteria. So a lot of people think that we've discovered every single species of bacteria. Well, that unfortunately is not the case. Most bacteria from an ecosystem, so fecal material or a lake or a pond, cannot be cultivated in artificial media, so in growth media. So what we use in microbiology is we use a lot of different augurs, and I like to use blood agar. And the bacteria, for a variety of reasons, does not grow. So before we had our techniques of gene sequencing like what we could, they were completely unknown to science. So I was able to take and to cultivate this species of bacteria. It's called an Enterococcus. It's not a disease-causing species. And I worked with the CDC. Even though it wasn't disease-causing, I still worked with the CDC, and they were really great. And they helped to characterize that and published a new species of bacteria a few years ago. So most bacteria are unable at this point to be cultivated, and almost every day scientists are finding new species of bacteria. So just this semester, I worked with a student. We were published a paper on the painted turtle, the bacteria associated, and we were the first group to do this. Nobody has looked at the painted turtle. When I got my the reviewer comments back in the discussion, the reviewer said, well, you need to put this more in line with other research with turtles. And my reply was, there isn't other research with turtles. It's sea turtles, but this is why I have to go with the discussion. And so the student and I, we found another new species of bacteria called a Clostridium bacterium. So we're working with another scientist and trying to publish that. In China, I work with the Yangtze finless porpoise. And before I started with this group, a really great group, there was nothing known about the microbiota. They didn't look at bacteria. They looked at reproductive status. They looked at mitochondrial DNA and population genetics, lots of great things. So the first thing that we found is that a couple of the Yangtze finless porpoises had Helicobacter. So Helicobacter pylori can cause gastric ulcers in humans. Well, this was a different species of Helicobacter, and it could cause ulcers in porpoises. So that's something that animals can be affected, the same as humans can be affected by disease-causing microorganisms.
So it seems to me there is a wild area of research just keep adding more bacteria from animals to a dictionary. So as your dictionary becomes bigger and bigger and bigger with the new technology, what's after that? Like a lot of people will say, well, that's interesting. We got new bacteria. Probably you receive this question a lot, right? And after that, where's the application? Why do we care?
Well, part of it is this. If you look in the literature, you look at a lot of disease-causing organisms. Okay, that's good.
About animal disease or
animal disease, human disease, right across the board. Okay, that's good. But what about the non-disease-causing organisms? How do you, when you want to compare, why does one organism cause disease? Okay, well, let's look at organisms that don't. So you can have more in the literature to take and to compare to. Another thing that's
Is it like a control group?
a control group, yeah.
You're
So
creating a control group for it, yeah.
you're creating a control group because most bacteria, it's less than 1%, cause disease. So you can take and you can see, okay, why do the bacteria cause disease as a control group? Also, microorganisms are very beneficial. So you can look, and there's been numerous studies in humans, the Human Microbiome Project, at different bacteria at different sites, and so many papers on the benefits of the microbiota. So how does bacteria benefit humans? How does it play a role that we didn't know before in disease? So that gives you an avenue of treatment. Also with animals, we care a lot about the health of animals, and sometimes there is transmission of microorganisms from animals to humans. Now that can be true in industries, but also that can be true in the wild. So an interesting thing that we found a little bit over a year ago is we were looking at the bacteria in giraffes. So we published that, and there's a class of antibiotics called carbapenems, and they're a very potent class, and we found a bacterial species in the fecal material of a giraffe that was resistant to this group of antibiotics. So why would it be there? You wouldn't expect it to be there because even though these were housed at the Jacksonville Zoo, they didn't get fed any antibiotics. So there's that whole component of transmission. Now, it's called a plasmid. Now, it's not the only way, but these small circular pieces of DNA can transmit genes, which make proteins, which allow bacteria to become antibiotic resistant. So you can find some very interesting things that you wouldn't have expected.
So basically, you're actually, if I imagine this, it's almost like going to a museum or a library with endless possibilities, and just go there, and every day you just find new stuff, like some structures you've never seen before, some properties you've never seen before, and based on that, you can make it to be like a disease, to think about potential threats to disease or antibiotics, or you can think about protections, such like the way we use microbiology. So
that's right, yeah. And then what's really nice about working at the technical college is I enjoy publishing, and I have the freedom to work on whatever I want. And I like to work on animals that people are not interested in working on. So such things as turtles. Not a lot of research goes into the microbiome of a turtle or a snake. So you can look at that. You can find interesting things like antibiotic-resistant bacteria, new species of bacteria, characterize the microbiome, and compare it to other animals. So it's a wide open field.
Interesting. So let's go back to your research method. Do you have actually a very standard or like a well-defined procedure? How do you start with a species and start to say, hey, how do I find some bacteria from it? And how can students actually participate in this?
What I do is I get the fecal material, and I send the material to a company called Mr. DNA. And Mr. DNA will take and amplify, this is called next generation sequencing, the 16S for bacteria. Now, if you're looking at fungi or other organisms, it's another gene, but it's a similar idea. And he will send me back those sequences and the identity to what's called the phylum level of the different bacterial species which are there. So I do that. I send that out to a company because I don't have the equipment to do it in-house. Now, in terms of growing the bacteria, really you can be very creative. What I like to do is I like to use a blood agar because it's an enriched media. So you can streak bacteria, just add the bacteria from the fecal material with a loop. So people can think of it like a pencil with a metal wire. You heat the metal wire, you get some of the fecal material, and just add it to an agar like Jello plate. And you can take and you can grow it in the presence of oxygen, so that would be aerobic, or in the absence of oxygen. So not every living organism requires oxygen. In fact, to some bacterial species, oxygen is toxic. It is poisonous. So in the fecal material, it's going to have a lot of bacteria that can grow well in the absence of oxygen. So you can just add that to a blood agar plate and see what grows. You can purchase selective media. So we purchased selective media to grow a species of bacteria called Bacteroides, just to see what was there, and we found some just interesting bacterial species. Bacteria that are in the Clostridiums, as well as other species, form these structures called endospores. Endospores are these dormant, resistant structures. So if you take like the cold, well, it's very hard for organisms to survive in cold like this. Not impossible, but hard. So bacteria which form endospores can survive in the cold. People think boiling water. Well, you boil the water, your kettle, the water's sterile. No, not necessarily. Endospores can survive the boiling of water. So what I do with my lab is take some of the fecal material, and the student and I, we put it in 70% ethanol. And you just tilt the tube, let it go for about a half an hour. The bacterial cells will be killed by the alcohol. Alcohol, 70%, kills bacteria, but it doesn't kill the endospores. So then what you do is you just take some of the liquid, you add it to a blood agar plate. You can incubate it aerobically, anaerobically, and you take and you pure culture it. So you isolate the different bacterial species, and I take the plates and I send them to a company called GeneWiz. What bacteria I have, I honestly don't know. That's the fun of it. You don't know what you've got. So just send it, and it comes back with this sequence. The DNA alphabet is 4 letters, A, T, C, G. So about 1,400 A's and T's and C's and G's. You get that, and you submit that to a government database called the BLAST database. Completely free, and within a couple of minutes, it matches your sequence to known sequences in the database, so you can take and identify your bacteria. So that's the protocols that I generally use. And sometimes if it's an interesting bacteria, you can sequence what's called the genome. So the DNA in that organism can be, send the bacteria again to Mr. DNA, and Mr. DNA sends me back little fragments. So it's like a big jigsaw puzzle. And you can take those little fragments, and there's at Argonne National Laboratory, there's a database called Patrick, and you submit those little 300-400 what are called base pairs sequences, and you look for the overlap. So there are computers, so they have a server room which would be about the size of probably pretty close, the basement's water cooled, and the top has got servers, so probably pretty close to the size of wing, just with servers. And within a day or so, they'll send you back your assembled genome. You know the different genes. You can identify the bacteria a little bit better than just based on a gene sequence. So we did that, and that's how we were able to show that one of our species was a novel Clostridium species. And it's all free, too. It's completely free.
Interesting. So after you actually discover something new, do you actually add your new bacteria into the database?
Yes.
They need to be evaluated, certificated by certain facilities or anything to make sure?
What we do is we sequence the genome, but you at least need the 16S rRNA gene. So whatever genes that you sequence, you send it to the NCBI, it's a government database, and they give you an accession number. So these accession numbers are included in your manuscript, so other scientists can use your data. You also have to take the bacteria and send it to a culture collection. So the American type culture collection is an example. So a certificate is sent from the culture collection. So culture collection will take your bacteria, they will sequence the 16S just to make sure it's what you said it was, and then they will make it commercially available so that other scientists can purchase your bacteria. So that's what you have to make available. And then what you have to do for a paper is you have to prove it's a new species of bacteria. So you need to do certain biochemical tests. What are the nutrients the bacteria uses, like different sugars, lactose, sucrose, glucose. You can look at the fatty acids. You can look at the gases which the bacteria produces. But you have to show that your bacterium is new. So you do all these tests comparing it to closely related species, and that goes for review. Then there's naming. So your name has to be approved. So the first name that I give with the Enterococcus, I called it Enterococcus horridus because it was from a timber rattlesnake, Crotalus horridus. So I wanted it sounding really horrible, Crotalus. And the journal said, no, it's not horrible. We're changing the name. So the naming convention also has to be properly Latinized, etc. So you can't just name it after yourself. You have to have the proper naming.
You probably have some rule to follow. They're going to send you.
They do. I actually sent it to, we were looking for a name, and we sent it to a professor, and he came up with a name. One of them didn't really like my name, said, no, I don't like it. Let's use it. Okay, fine. We'll use this name. So it really is, I like doing it, but it's a lot of work to actually prove your case that this is a new species of bacteria.
It sounds great because you basically have a very well-defined process. You first do the first screening with the database and see here's the potential. Now you grow it and try to measure the property and try to prove it. And you have a verification process. You have everything basically settled. So for me, it's almost like a factory, right? You have a business process defined. Now you just need to have the patient and you have a little bit of luck.
That's right. A little luck. That's right. And that's the nice thing about using animal samples is because they're very diverse. Nobody's looked at them. So in animals which are plant eaters, it's been shown that the bacterial diversity is huge, much, much greater than animals that just eat meat. So I'll use any animal species, but something such as a giraffe, it's going to have a really diverse population of bacteria, and there's just a lot to find if you're looking in places where other people are not.
That's actually where I want to ask next. How do you find a potential good sample to begin with? Like what kind of animals are you looking for, or how do you have this inspiration of what plant I want to collect?
A lot of what I do is I like to work with animals from the wild. Now, I'm not opposed to zoo animals. I'll work with that. Part of it is a collaboration. It's just very difficult to get this. If I know a person who works at a zoo, I'll email that person. Could you give me some samples? Sometimes I'll just email somebody, and that's how we got some blue whale samples. So we got a couple of blue whale samples from the Pacific Ocean.
Oh, okay. Yeah. I'm wondering, there's no zoo.
No zoo there. No zoo here. Anna sent me 2 blue whale samples. So we were
But if I could interrupt, how do they do that? Like,
they go out on the boat and they can spot a blue whale, and then they use dip nets when they see the fecal material, and then they collect the fecal material with the dip nets, freeze it, and send it.
That's crazy because these guys, they just poop in the
that's
ocean.
right.
That's you go there and find
that's
something.
Right. That's what they do. So it's not so easy to always find this. And I worked with a person from UW system, a whitewater in the turtles. So I just he was interested in turtles. Emailed him. When you're doing your study on painted turtles, could you collect some fecal samples for me? And they said yes. Right now I've got some African wild dogs from a zoo. So I do a lot of work just looking for what's out there, and just trying to trying to make a contact.
Is it purely random, like I need to see whatever is available, or actually you kind of have a hunch, like, yeah, this kind of species is more likely to happen? And also, what's the success rate if you complete this process for 10 times? How big the chance you can discover something new?
Well, in terms of, I'm willing to, because there's so many animals that have not been characterized. It's really a very wide open field. So I'm open to any type of animal. So I don't really have a hunch which one's going to be, and I'll stay completely away from animals such as cattle and pigs and things like that, because commercial, because there's way too much competition that's been done. So I go with what hasn't been done. In terms of finding a new species, that's really a hit or miss. I was trying to find a new Bacteroides species. I didn't find one. So maybe 1 out of 10, you're going to find it if you're lucky. Yeah, it's a hit or miss. So there are labs which are really designed for that. My lab isn't quite that. So if I happen to find something, that's great.
Interesting. That's fascinating to see, and especially in the process. I also hear there's a national database about this, which I never know of before. So it seems like you have the right company doing the right thing. So it's a very, very sophisticated process with different industries involved. So that's great. So which part you usually involve the students?
I involve the students in taking the fecal material and isolating the bacteria. So we will do that. Then they will send it to GeneWiz, get the sequence. We blast that. If it's worthwhile, we sequence the genome, and they work with me on sending the data to what's called Patrick at Argonne. And then we just take and we collect our data. And in terms of writing the manuscript, we spend a lot of time working together, writing our manuscript with all of the data that has been collected. So what I do at Gateway is it's 15-week semester, and you've got about 12 weeks to do the research. So there's enough time, but not a lot of time. So mostly growing the bacteria, analyzing the data, and then in the writing of a manuscript.
Good news is actually just growing the bacteria may take some time. So
sometimes, yeah.
Spent on waiting, right?
That's right. Sometimes you have to wait a week or more, and sometimes too you just all of the sending and receiving of the data can take some time too. We send our bacteria out for antibiotic resistance. There's a laboratory, Marshfield Laboratory, which will do isolates from animals, looking at that. So over the years, just a system, as you say, of different companies and different techniques, which seem to work quite well for the Gateway students.
That's pretty nice, yeah. If I imagine I'm a student, I'm basically involved in everything, the whole process, and if I really put my effort into it, I will be able to do this when I graduate, right?
Yeah,
at
absolutely.
At a certain level, and then that's become future collaborations. Now you have more people doing this, and eventually the library just grow bigger and bigger and bigger and bigger, and something interesting gotta happen after that.
That's right. So, that's right.
Actually, a side topic, which I didn't put in my question list. I talked with Scott Cooper in my second episode, and he was talking about sending some kind of bacteria, the fungus, or to Mars. Have you discovered any bacteria which will actually survive the harsh environment in outer space, and maybe sending them there will benefit human in some way?
I have not. What I look at are mostly bacteria which grow regular. There's different domains, and Scott talked about that. In the domain a they grow at body temperature 37 degrees Celsius at a near neutral pH. The archaea would be something that you would find at hot springs, in geysers, etc., which can survive above the boiling point of water and undersea thermal vents. So that was a completely different area of looking at those organisms. So I look at the more moderate organisms, nothing that's going to survive any super harsh conditions.
Interesting. Okay, so let's talk about your work on the Yangtze River poppers. And how did you? I know you're personally involved, not just your research. Your family actually got whole involved in this. You traveled to China. You actually got involved in a lot of not just research activities, right? Like promoting to save or protect the mammals. How did you get started?
I got started because my wife's family knows the director, Dr. Wan Ding, and he works with the Yangtze finless porpoise. So I go there during the summer, and I wanted something scientific to do. So my wife set me up with him, and they did a lot of great work, but nothing with the microbiota. And just to give you a little background about the porpoise. So it started before there was a white dolphin, the baiji. And the institute originally started to save the baiji, the white dolphin, which was part of endemic in the Yangtze River. However, that effort did not go well, and it's believed to be extinct. And it's extinct.
When I was training my elementary school, where we still put that in our textbook, but within 20 years, it's gone.
It's gone. It's completely gone.
That's sad.
Yeah, it's completely gone. So they did an expedition years ago, looked for months in the Yangtze River, and they found none. So it is extinct. So they said, okay, let's look at the Yangtze finless porpoise. So the Yangtze River, as you know, is very important in terms of transportation. There's just and cargo, billions of tons of cargo every year, factories. With that's really great for industry and fishing. There are problems for the animals which are living there. So there can be acoustic pollution, the noise of motors, motor strikes, the propeller hits the animal. Chemicals which are placed in the water. Fishing with gill nets can actually take. So a porpoise is a mammal. So like us, we have to breathe air. Well, so does the porpoise. So the porpoise can stay underwater longer than we can, but eventually it has to come up for air. If it's trapped in a gill net, it drowns. So these animals end up drowning. So there is an oxbow lake called Tianzhou, which in 1972 became separate from the Yangtze River. So Professor Wan Ding said, there's a lot of problems right now with the Yangtze River. Why don't we take these porpoises and excite to conservation, remove them from the Yangtze River and put them in this oxbow lake? Not a lot of people liked his idea. I
can imagine.
Yeah, it was, and to be honest, he didn't really love his idea. But he said, well, let's look at the Yangtze River. The conditions are not great right now. Let's move them to better conditions and then work on fixing the conditions in the Yangtze River. This was the first example of ex situ conservation of a cetacean species. It worked super, super well. If you look at pictures of the Yangtze finless porpoise there's a dolphinarium where I work. They look, really lean. They're in good shape. They're in the reserves. They're even in better shape. They're overweight in the reserves compared to because we don't overfeed them. So the success of the birth rate is really, really good because there's fish there, there's no pollution, there's no fishing, there's no anything to disturb them. Part of the problem is how many porpoises can you hold in this oxbow?
Is this reserved artificial?
No, it's natural.
Then do we worry about the balance at all, the equal balance?
At this point...
You're just aiming at one particular species, they can have a good life, but what about all the others? Back in the reserve.
That's I think at this point what they're looking for in that artificial it's like 21 kilometers. So it's not really huge. So it's big enough for the porpoises, but I think they're looking at these reserves. Let's keep the porpoise population stable because right now there's about 1,000 porpoises in the world. So it's considered critically extinct. So let's just keep that population stable and then think of ideas of what we can do to return porpoises to the Yangtze River, clean the river. So industries can function. People could fish or do other industry.
So basically a sustainable plan.
A sustainable plan for the porpoises. So that has worked really well. What Wan Ding has done, he's done a lot of really great things.
And this was done during the past, or 5, 10 years?
It was started in the 90s. In the 90s. So it's been for a number of years, and it's one of the things that is happened is we've had success in the dolphinarium in both artificial breeding and just through natural means of porpoises having offspring. But what sometimes happens is the offspring dies. So it's not always 100%. So humans will give IV or antibiotics, and sometimes the porpoise survives, sometimes it doesn't. But in the wild, it seems to be better because humans don't intervene. So it's a matter that I was told by a scientist, the most important thing is mother-calf bonding, because you need the milk. And scientists don't always know in the dolphinarium if that bonding is occurring until it's too late. But in the wild, there is no other choice. Humans are not going to intervene, so it seems to work. It seems to work better in the wild. But I will say, we had a successful birth of a porpoise. I was there in late May, June, and I was there for the 1 month. We had cake and celebration of the birth of the porpoise. He's doing great, and they had the 100th day celebration of the porpoise. So we had a baby porpoise, male porpoise, who born just this year, and he's doing great.
Awesome. That's really awesome. So you started with this, and now can you tell me a little bit about what you do there?
Sure. What I do is I work with Professor Jin Song Jun, and he is in charge of genetics. So he has graduate students, and I supervise the graduate students. So what I did with one graduate student this year, Leisha, is this isn't from the Yangtze finless porpoise, but this was from a whale. We found a bacterial species in the abscess of a whale, and we took and we characterized the bacteria associated with the that was present in the whale. So I do a lot of work with the graduate students there. I get the graduate students looking at the microbiota. We started with the Yangtze finless porpoise. So we looked at the fecal material of the Yangtze finless porpoise, and you can look at that at the dolphinarium and compare that to the wild. So we've done work on.
What's the conclusion there? The difference.
There are some differences, but if it's a nice clean environment, the similarities are stable. So we just more or less we just took an inventory. We just want to see what's there, and then you can take and you can figure out, well, what can we do after we have an inventory? Do we see any potentially disease-causing organisms which are present that we have to worry about? So for some animals, there are vaccines. So brucella is a problem. So should we vaccinate the animals? Should we supplement their diet with maybe some type of probiotics, something like that? So we can think about that in future. So right now we're doing inventory, seeing what's there, comparing differences, and then in future we can plan what we can do with the information that we have.
So in the big picture, you're helping the whole reservation from a certain perspective, but you work with, but there will be other people working on all kinds of perspectives to see.
All kinds of perspectives. Yep.
What's the scale of this project? Like the whole thing, like put them in the reserve and then do what you need to do with Yangtze River and put them back. Do we have a timeline?
I do not know of any timeline, but I'm not involved. There are people who work with the Chinese government, both local and federal, about that. They would know much more of the timeline. I'm not really aware of what the government is very favorable towards this, what the different exact steps that are being done are. The Chinese scientists, though, would know much more. But I know the government is very interested. They're very favorable to this. I told the director of the institute, he laughed, but I told him he should invite Xi Jinping to come and have a look at our what we do. And I think I was kidding, but I was also very serious. Is that I think he would be very impressed at the fine work that these scientists have done. So the idea of public awareness, that's something that I try to do. So there is a school that is kind of an ambassador for the Yangtze finless porpoise. So my daughter and my wife and I, we went there and we talked to the students there and presentations on the Yangtze finless porpoise.
How old is Jessica when she was doing it?
She was 10.
10. Okay.
So she went a couple of years ago and took some pictures and met the students there. So a lot of education of people in China, because not everybody knows about the Yangtze finless porpoise in China. So that's a great opportunity to get school children involved, and then a broader picture. And it was a couple of years ago, so education is really important, that a princess from the UK, Princess Anne, she came to visit, and she spent about an hour or so. So Jessica got to meet the princess, and she was very interested in the conservation efforts and the porpoise and what we're doing. So nice to have advocacy like this. There's been Olympians that have attended. There's a very famous actor, I won't pronounce his name correctly, Putanshi.
I have no idea, but I think I can Google it.
Yeah, Google Putanshi. He's a very famous movie star.
Oh, yeah. He's older.
Older man, yeah.
He's older, yeah. Okay,
yeah.
I see, yeah.
Yeah, Putanshi.
Putanshi?
Yeah, okay. Yeah, I told you I'd mispronounce it. I don't pronounce any Chinese word correct.
I think, yeah, that's him, yeah.
So he is an ambassador for the porpoise, and he actually came. This was his first time there, and my family and I got to meet him, and he got to see the porpoise. I gave a talk at Jessica's elementary school on the porpoise here in on Alaska, and I also got the opportunity to talk to people at Gateway Technical. So we did a joint project with bacteria. So in China, they talked about sequencing the genome. They sequenced the genome of the bacterium, and then using the database, the Patrick database here in America, and it's really available too in China, but I did the project here with a Gateway student, learned about the porpoise, made a presentation about the work that we did in America and in China. So public education, I think, is really a big key to this, and the more the public becomes interested, I think the more things will be done.
It's amazing to see, like, this is actually what we call the value of international cooperation, right? International collaboration. It's like some people in China collect the stuff, and then they do some research, they send it your way, and within your process, and you can actually get a sequence and get back to them, and it's amazing, like, how many people got involved, plus all the relationship you build towards it during this process. So it seems to me, not only you do research on stuff you love, you also got personally involved on the non-research side. It's almost like a whole package.
Yes,
right?
yes.
It's like a lot of people do research, but they don't get involved in what's after the research or what's before. How does that change your personal perspective on research or your life in general? Because I feel like that must have some impact, maybe.
Yeah, it does. I think if you really like something, then you're going to be more passionate about it, and you're going to enjoy it more and more. So I think for me, just the idea of the public awareness and how can we take and talk to the group about and I'm very willing to give talks and encourage them to give talks, and they do give talks to elementary school students, and all the way up to government officials to make people aware of the Yangtze-Finless porpoise, the population decline of the porpoise. So there's still time. That's the good news. With the porpoise, there's still time. So there's the vaquita off the coast of California that there's only about 30 of them left. So unfortunately, time is running out, and maybe it won't be possible to save that. But if we act, and if we act in a responsible, reasonable manner, then this species can be repopulated. It can be saved. It can not go extinct. So I think just the role in education and public awareness and advocacy is what I like to do.
It's interesting to see with the economics going on, all the bigger pictures, but people are still paying attention to very special things. I'm going to play the devil's advocate here. Like, what if some people just ask you, why bother? Like, why bother with all the dying species with so much resource? What will be your response?
Well, my response is that every species has value. So there's value there. The Yangtze-Finless porpoise is an indicator species. So it's at the top of the food chain. So in the Yangtze River, if the top species is doing really, really well, then you can assume everybody else is doing really, really well. But if the top species is struggling, which the Yangtze-Finless porpoise is, then so is probably every other species. So it's an indicator that there's more problems than just with the Yangtze-Finless porpoise. So the Yangtze-Finless porpoise doesn't just live by itself. It lives in an ecosystem. So it needs clean water. It needs water. It needs a food source. Well, that, which is mostly carp, but that carp needs a food source, etc. So if your ecosystem is not doing well, then, and it reaches all of the top level, that tells you that your entire ecosystem is going to be in a lot of trouble. So it's an indicator of more serious problems.
So it's a signal the system is sending you to say, pay attention.
That's right.
Is it also fair for me to say, if you can fix this, maybe you fixed everybody below that?
That's right. So if you fix that, then you're going to take and you're going to, because it's an ecosystem, it's like a food chain, a food web. Different species depend on each other. So if you fix that, you're going to have a healthy, diverse ecosystem, which is beneficial for the aquatic environment.
That's very convincing to me. Like, at least to me. Like, I think, so you're not just seeing one species or like just out of empathy or anything. It's actually, it's the first step to dive into some deeper problem, and you can only know more until you start the first step, and then you figure out, okay, one thing leads to another. Now I know where the root cause is, and then start doing something about it. Interesting. So, do you think the idea you talked about was really interesting? That is, put some species to a reserve, do whatever you need to do, and do the study at the same time, because once they're gone, you won't be able to do the study anymore to figure out the cause. Do you think this thing is replicable?
Oh yeah, absolutely. There's a number of different reserves where they're taking and they're putting the Yangtze-Finless porpoise in that particular reserve. So yes, there isn't just a Tiana Joe reserve. There's
But what about like two other species, like who are facing the threat of extinction?
I think that there, to some extent, yes, that this ex situ conservation is, you're able to replicate it, but an animal such as the vaquita, it has not been successful because
have they done that?
they've tried, but it's a very shy species and it just doesn't do well. So for some animals, it's going to be, you're going to take and you're going to be able to replicate that. But for other species, you need to start sooner and fix the problems in situ. So in the area where they're living. So sometimes ex situ, okay, it works if in the area is not going to work, but ideally you want to fix the problem in the area where they are living because that's where they're living. So for some animals it is, but for others not.
And maybe we should also consider the symbiotic relations between different species. Sometimes you just move one, you didn't realize there are so many more you need to actually start thinking.
That's right, yes. And there's also, there's a very big aquatic, it's called, and I'll mispronounce it again, it's in Zhuhai, it's called Chalon.
Chalon, maybe.
Yeah, Mal will pronounce it correctly for you. And it has the world's largest aquarium, and there's whale sharks in that aquarium.
Wow.
So yeah, when you're in China, you can take your family.
I never realized how big that thing is until my friend sent me a picture. Actually, she was in Hawaii and swimming with that thing.
Yeah.
And then I see, okay.
Yeah.
We are side by side. I know how big that thing
Yeah.
is. It's
yeah.
crazy.
It's the world's largest aquarium, and there's talk about putting the porpoise in Chalon in an aquarium there because it's huge. So again, that it could be an environment where the porpoise can thrive and people can learn about the porpoise and become aware of that. So that's another approach, putting it in a commercial aquarium like that.
What's your opinion on aquariums? Like, should we even be doing that, or zoos in general?
I think zoos are good as long as they treat the animals really, really well. So such things as the San Diego Zoo. There was a person, Chuck, that came and talked about the zoos and aquariums at the Institute in China. So he's from the San Diego Aquarium, etc. I think it gives you an opportunity to study the animal, to learn about the animal in ways that you would not, come up with strategies to reintroduce the animal back into the wild if the animal can be reintroduced. So such things as Ocean Park, San Diego, they're very clean, they're very good. Chalon, which I'm talking to you about, it's very nice. So I think for the animals, it does have benefit.
So, I think we're hitting the time mark here, but I want to close with a philosophical question. What's your vision within the next 100 years, the relationship between human and other species?
I think that I think it's getting better and better. Certainly there are problems, but public awareness is growing with such things as podcasts and internet and television. With new technology, we can take and we need to produce. We need to have industries, etc., for our daily lives, but we can produce in such a way that there's less pollution and there's less impact on animal species. So I think with growing public awareness, with good public policies, with new technologies and future technologies, that we can get along with the animal species very well and let them be animals, don't infringe on them, and at the same time, we don't lose our quality of life. So I think with the right steps and the right legislation, it can be a very bright future.
Do you think it's a win-win? It can be a win-win situation, or we need to sacrifice a certain quality of our life in order for that to happen?
I don't think that, honestly, people are going to sacrifice. And if you ask them to, I think
it has to be a win-win.
It has to be a win-win. I just don't think people are going to say, yeah, I'm going to give something. They're not going to give anything up. So I think it has to be a win-win. And I think that it can be. It can be
Do
a
you
win.
have a successful example of win-win situation, even just for one species in a very small area?
There's the peregrine falcon that was almost extinct, and that has come back and repopulated very nicely. There's the bald eagle, the eggshells, DDT. We no longer use that. That's come back, and that is repopulated very nicely. I think the steps that we're doing with the Yangtze finless porpoise, we're not quite to repopulating, but I think we're certainly on the right direction, and I think the future is bright.
Yeah, mathematically, you're not increasing, but at least you hit the local.
Yeah, we hit we hit that, and
Make
we
it stable, and then
make it stable. We start it soon enough that that there is time, and we've got some things in place. Keep the things in place, and work on the Excite2 part of it, and come up with strategies there. I think that it's going to be possible.
Cool. Thank you so much
All
for
right.
the talk today.
Thank
that was
you.
really educational for me. Thank you.
Thank you.